Welcome to the CJD Foundation's virtual conference with tonight's speaker, Dr. Ann Smith of Ionis Pharmaceuticals. This is Debbie Yobbs for the CJD Foundation. We've watched with interest the progress toward applying antisense oligonucleotides to prion disease. In 2016, thanks to your donations, the CJD Foundation was able to award a grant that was one of the funding sources for a project by Byron Coey at Rocky Mountain Labs, and a team that included Sonia Valab and Eric Minikel, titled Antisense Oligonucleotides to Delay or Prevent Onset of Prion Disease in Mice. Then in 2019, your donations funded another CJD Foundation grant related to biomarkers and ASOs with Dr. Stephen Arnold at Mass General. You can learn more about these studies on our website. Your donations to the Foundation can continue to seed the ideas and discoveries of tomorrow. To learn more about the work of this year's CJD Foundation grant recipients, join us on October 22nd and October 28th for the last two webinars of our virtual conference series, featuring 7 of our 9 grant recipients. If you missed the first panel with Holger Ville and Sonia Valab, you can view it on our website. If you have questions during the presentation, please enter them on your screen and we'll answer as many of them as we can following the presentation. And now, to introduce Dr. Ann Smith of Ionis Pharmaceuticals, is Dr. Brian Appleby, Director of the National Prion Disease Pathology Surveillance Center and the Medical Director of the CJD Foundation.
It is my pleasure to introduce Dr. Ann Smith, who is an Executive Director of Clinical Development at Ionis Pharmaceuticals, a California-based biotechnology company focused on RNA-targeting therapeutics. She is responsible for shepherding promising therapies from the research stage through clinical trials. She specializes in neurology drug development and leads teams working in multiple disease areas, including prion diseases. Many of you are aware of the work that several researchers have done on antisense oligonucleotides for prion disease, and Ann will be continuing this discussion by presenting on antisense oligonucleotides as potential treatment for prion disease. Thank you for joining us, Ann.
All right, thank you to the Foundation for inviting me to speak about Ionis's prion disease program. I have to say I was really looking forward to attending the conference this year. I have colleagues who've gone in the past and they returned to the office genuinely moved and motivated by the people they met, the families, the researchers, the Foundation leaders. But for this year, we'll do the next best thing and meet virtually. While the pandemic has impacted each of us in many ways, I'm happy to report that the prion team at Ionis hasn't skipped a beat. Many of us are working from home, but the work hasn't stopped. So I'm going to talk to you today about Ionis's prion disease program, and let's get right into it. Really quickly, Ionis is a pharmaceutical company founded in 1989 for the express purpose of developing a discovery program for a new type of drugs. These are drugs that target RNA molecules, which I'll talk about in a few minutes. This effort required an extraordinary dedication to the science, to making discoveries in the underlying biology, making advancements in medicinal chemistry, to turn this idea into actual drugs with properties needed to work well in the human body. We've published extensively on these advancements, and we now have 4 approved drugs in this class and more than 25 drugs in clinical trials for a range of conditions. We're located in Carlsbad, California, and we're relatively small by pharma standards at about 500 employees, with expertise from drug discovery through research and clinical development and manufacturing. I want to tell you how Ionis's drugs work, but first let's talk about what they are and what they're not. You can think about drugs as falling into 4 different categories. There are small molecules, protein-based drugs, nucleic acid therapies, and gene therapies. And the category that Ionis has pioneered is the third one, the nucleic acid therapies. But let's talk about each of them quickly. For small molecules, think pills in your medicine cabinet. These are the oldest and by far the most prevalent type of drug. Bare aspirin's been around since the late 1800s. Approximately 90% of drugs on the market today are small molecules, so this is the really common class. They're easy to make, they're easy to use, they're easy to store. Often the precise way that a small molecule works is not known, and it's not uncommon to have side effects because the drug might act in multiple ways, in non-specific ways, but nevertheless, there are loads of really important small molecule drugs. Protein-based drugs came next in the 1970s and 80s. The biotech era kicked in, and We figured out how to manufacture proteins. The platform started with protein replacement therapies like insulin or human growth hormone, where companies made the protein to be given to people whose bodies weren't able to make enough on their own. Then came what are called monoclonal antibodies, and since then it's branched out, and now there are hundreds of protein-based drugs in the market. You've heard of a lot of these brand names like Enbrel and Herceptin. Protein-based drugs are more difficult to manufacture than small molecules, and they can be difficult to store and difficult to use, but there are many good, important protein-based drugs on the market. Nucleic acid therapies and gene therapies are the newest types of drugs, and as I said, Ionis's drugs are antisense oligonucleotides, or ASOs, also called antisense drugs. Sorry, we have a lot of different words for the same thing. But those are all in the nucleic acid category. And as a platform, ASOs have very desirable properties. They're easy to make, they're easy to store. They're also highly specific, which means that we know where and how they'll act. We've designed them in a very deliberate way. And I'll leave it at that for now, because in the next few slides, I'm going to explain how ASOs work. And I'm also going to talk in a bit about how gene therapy drugs are different. So just for now, understand that they are different. So a little bit of biology. To understand how ASOs can be used to treat a disease, you need to understand this one biological relationship, that DNA codes for RNA, which codes for proteins. And you probably remember DNA from high school biology. DNA always got the spotlight. It's your genetic material. There's a copy of it in each cell, and it stores all the information that makes you, you. It is incredibly important, but let's let proteins share a little bit of the spotlight today. DNA is the genetic material. It's what's storing the recipes for proteins. Genes are recipes for proteins, and proteins are incredibly important. Most of the work in your body is done by proteins. They're required for nearly everything a cell is and does, including its structure, carrying out chemical reactions, transmitting signals, binding to viruses to protect the body. In a nutshell, life is dependent on your ability to make the proteins you need in the quantity you need them at the time you need them. And it turns out that a lot of diseases are caused by too much or too little of a particular protein. So the DNA, the recipe book, is stored nice and safe and sound in a special protected compartment in the cell that's called the nucleus. And when your body wants to make a protein, the DNA will unravel just a little bit to expose the stretch, the gene that is the recipe for that particular protein that's needed. Then the cell makes a copy of the recipe. The copy is called RNA. And the RNA leaves that protected area in the nucleus and goes out to the cell, where it encounters what are essentially these little machines called ribosomes that read the recipe and make the protein. So you can imagine that if there's a disease that's caused by too much or too little of a protein, there are a number of different approaches you could take to treating that disease. Let's talk about prion disease specifically. We know from experiments in animals that if you can eliminate the prion protein or knock it way down, the disease can't take hold. So prion disease is a situation where we'd like to find a way to lower the amount of a particular protein, the prion protein. So how do we approach this as drug developers? Well, looking at this slide, you can see there are a number of different targets. We could target the protein itself. We could look to identify a small molecule or a protein-based drug like an antibody that binds to the prion protein and causes it to be degraded. And there are people working on this. If they can find such a drug that gets into cells, reliably attaches to the prion protein, causes it to be degraded, and doesn't inadvertently attach to other proteins or cause undesirable side effects, that would be a great drug. Another approach would be to target the DNA itself. Start at the beginning. We know that the inherited forms of prion disease are caused by small errors in the prion gene, and those small errors cause the resulting protein to be more susceptible to misfolding that occurs in prion disease. So if we could get into the nucleus of every cell, get to the prion gene itself, that little stretch on the long piece of DNA, and fix that error, that could also be a great drug. And that, by the way, would be gene therapy. Gene therapy is a drug that works directly on the DNA itself, either introducing a new piece of DNA or changing on the DNA itself, that recipe book itself. The final option is in the middle. That's where nucleic acids come in, like Ionis's ASOs. We target the RNA, we target the message. ASOs can be designed to work in a variety of ways, some of which will cause an increase in the amount of protein and others cause a decrease. But again, in prion disease, we want to decrease the amount of prion protein. So what we can do is design an ASO, a little strip of material that kind of looks like DNA or RNA, that binds to the RNA for prion and causes that RNA to be degraded. If there's less RNA, the message, the copy of the recipe to make prion protein, then less protein will be made. So antisense drugs are specific, titratable, and reversible. They're specific, which means that we can design an ASO that will bind only to the RNA that codes for the prion protein. The ASO won't bind to any other RNA for any other protein, so we have confidence that we're minimizing undesirable side effects. ASOs are also titratable, which means if you can deliver a small amount of ASO, you'll get a small reduction in the amount of protein that's made. If you deliver a large amount of ASO, you'll get a large reduction in the amount of protein that's made. So you can fine-tune how much destruction and how much protein. This is important in some situations where too much of a protein causes one disease, but too little of that same protein causes a different disease. In that situation, you want to dial in the magnitude of the effect just right, but fortunately, we don't have that issue with prion disease. There are no concerns with knocking down the prion protein. And then finally, the effects are reversible, which means that if you stop giving the ASO, the effect stops. Practically, what this means is that ASOs are treatments, not cures. We're not manipulating the DNA itself. We're not changing the message that's been being made. We're just catching it before it gets to the ribosome and can make a protein. If you want to keep the level of a protein suppressed, you have to keep delivering the drugs, like a lot of chronic treatments of drugs. So this slide just shows that antisense therapies can be useful in all areas of the body, really in any condition that's caused by too much or too little of a protein. ASOs are a viable treatment strategy. And one note about ASOs for neurological conditions. They don't cross the blood-brain barrier. So you have this protective layer around your brain and spinal cord that serves to keep toxins out of the brain, which is pretty important day to day, but it does get in the way of making drugs that need to get to the brain. It makes it a little more complicated. So we solve that with our drugs by delivering them intrathecally. This is an injection in the lower back into the fluid that runs along the spine and surrounds the brain. For women out there who've had epidurals, an intrathecal injection is a similar sort of procedure to that. We have a lot of experience with intrathecal delivery. Over 5,000 patients in several different conditions have received our drugs intrathecally, and the procedure is really very well tolerated. So let me show one quick slide on the drug development process before we get into the data for our Prion program. Drug development can loosely be broken down into these 5 steps shown here. In the research and discovery and preclinical testing steps, we're thinking through the drug design strategy. What do we want to target, when, how? For prion disease, we want to target the prion RNA with the goal of lowering it so that less protein is made. For the how, we're using an antisense drug that's specifically designed to bind to the prion RNA and in doing so, destroy it. The when is a more complicated question, and you'll see we've done experiments to understand when in the disease to intervene. In this stage, we design ASOs and put them in animals to see if they do what they're designed to do. Then we go through a rigorous process to design the best ASO for human testing. So let me stop and let's think for a minute about the relative sizes of RNAs to ASOs. The prion RNA has thousands of nucleotides. Nucleotides are the building blocks of DNA and RNA and ASOs. Think of them as beads on a chain. So the prion RNA is thousands of beads long. And an ASO is 16, 18, maybe 20 beads long. So if you think about where you want to put the RNA, where you want it to bind, put the ASO, where you want it to bind on the prion RNA, and you walk along those thousands of nucleotides, there are literally thousands and thousands of possibilities of the ASO to design to bind to the prion RNA. So it's a pretty big task to consider all those possibilities and select the ASO that's most likely to be potent and well tolerated in humans. So that's one of the big steps of this stage. But once we've made that selection, that human candidate drug is put through a series of animal studies called toxicology studies. That's a standard set of studies that regulators would expect you to do, mostly to test the safety. And then we can move on to clinical trials, where the objective is to test for safety and efficacy of the drug. So broadly speaking, trials can be described as phase 1, 2, or 3, where the early phase studies are in small numbers of people, and the objectives are usually just to test safety and to learn about how long the drug stays in the body. Then the later phase studies are in larger groups of people, and the objectives shift more toward testing for signs of benefit. That said, the distinction between phases often blurs in rare conditions like prion disease. It's common to combine phases or even skip phases. The burden on the drug developer is the same regardless of disease. We must demonstrate that the drug is safe and effective, but there's a bit of freedom to be more creative about how you get there with rare diseases. And once we've generated the data that we believe demonstrates our drug is safe and effective, we can meet with regulators like the FDA to see if they agree. If they do, then the drug can be marketed. And our obligations don't end there. We have an obligation to monitor marketed use of the drug and react to new information as it becomes available. For the Prion program, you are here. We're in the final stages of tox studies with the Antisense drug that we want to test in people. And clinical trials are the next step. So let me walk you through a few slides of data that show why we're so excited about this drug as a potential treatment for prion disease and how we're gearing up to start those clinical trials. The major questions for drug design and animal testing were, can we design an antisense drug that reduces the amount of prion protein produced? And do animal models of prion disease benefit from this reduction? So remember the relationship. DNA codes for RNA, which codes for protein, and the protein causes the disease. If we have an animal model of prion disease and we introduce an ASO, does the ASO work? Does the amount of RNA decrease? Does the amount of protein decrease? And are the signs or symptoms of disease in that animal model reduced? And then importantly, can we do this safely enough to give us confidence that it'll be well tolerated in humans? Answering these questions was years of work involving tests in test tubes and rodents and monkeys. We have a great team of researchers at Ionis, and we have had the pleasure of working with outstanding collaborators in academia to build this body of evidence supporting the use of an antisense drug in prion disease. I can't begin to do it justice to the volume of work in this talk today, but I do want to show you at least a little bit of data. Using mice to model human disease. So it's really helpful in drug development if you have what's called a mouse model, meaning you have a mouse that gets prion disease or something that reasonably approximates prion disease. Unfortunately, in this area, we have a pretty good one. For these experiments, you start with tissue from an animal that had abnormally folded prion protein, like the human condition. This is a sick mouse. And you inject some of that tissue from the sick animal into a healthy mouse. What follows is a period of time where the disease builds silently. The animal doesn't have any outward symptoms of disease, but we can see in the brain that prion proteins are misfolding. And then there is a threshold that's reached, time point where the mouse does start to display symptoms. So if we zoom in on the brain tissue from the mouse, we can see in that pink picture at the top what look like white holes with little arrows pointed to them. These are called spongiform degeneration or vacuoles, and they are just openings that you wouldn't expect to see in tissue. And in the picture on the bottom, sorry about that, in the picture on the bottom, we see brown patches that are clumps of prion protein. All animals make prion protein, all humans make prion protein, but it shouldn't clump like that. These two findings are very similar to what we see when we zoom in on brain tissue from people with prion disease. So these are humans now on the right. We see the same abnormal white vacuoles, and we see the same abnormal brown clumps of prion protein. So there are satisfying similarities in the pathology in the mouse compared with the human. The spongiform degeneration, the white spots look similar between mouse and human. The prion deposition, the brown spots, the clumps of prion protein also look similar. And there are other similarities we've listed here that I haven't shown. Also, the mouse have an outward appearance and a progression that mimics the human disease course pretty well. They have rapid progression once symptoms appear. They lose motor skills, lose body weight, and die early. You can't lose sight of the fact that they are indeed mice. So I'll note that there are some differences too. Most notably, the incubation period is very consistent in mice. From the point of infection to death is usually around 4 months, whereas the incubation period in humans is quite variable. Also, the mechanism of infection is different. With mice, we're injecting them with misfolded prion proteins, while the human forms of prion disease usually involve spontaneous misfolding or a mutation that caused some susceptibility to misfolding. All that said, as mouse models go for disease, this one really is pretty good. So we use this model to ask important preclinical questions. First, does the ASO that's designed to target prion RNA actually knock down the RNA and result in less protein being made? And the clear answer is yes. These are data in mice showing the amount of prion RNA on the left and prion protein on the right. And on each plot, just follow the lines. The dots are individual animals, but the lines show you the trend. Where the green and blue are two different ASOs that were designed to bind to the prion RNA, and brown is a control ASO that's specifically designed not to bind to the prion RNA. So with the blue and green ASOs, the more ASO you give, that's the bottom axis, as you move from left to right, is a bigger dose. The more ASO you give, the more you knock down the RNA on the left, and the more the protein is reduced on the right. So it looks exactly as we'd expect it to look. Then when we look at the tissue itself, we knew that the mouse brains showed those two obvious signs of disease, brown clumps of prion protein and white vacuoles. So when we looked to see if the ASO treatment could help prevent these findings, this is what we found. On the top row, we have a healthy mouse that's not been injected with misfolded protein. So on the left, you see a lot of brown, but it's not clumped. That's what we'd expect. Healthy mice, just like healthy people, make prion protein. So it's there, but it's not clumped. So in the healthy mouse, we see the brown prion protein scattered around and not clumped. But on the right, we see a purple picture like we looked at before, and there are no white spots. There are no vacuoles, no evidence of spongiform degeneration. So this is what healthy tissue looks like. In the second pair of pictures, the second row, we have a mouse that has been infected with prion protein. I didn't mention it before, but it takes about 120 days to start to see symptoms in this model. So here we're looking at the brain of the mouse after 146 days. This is a mouse that had obvious clinical symptoms of disease. And when we look at the prion protein, we see brown clumps. The protein is definitely misfolded and formed these clumps, also called deposits. In the picture on the right, we see the white spots. These are vacuoles. So just like on the earlier slide, if we infect a mouse with misfolded protein and then wait a while, we're going to see prion protein clumping and vacuoles forming. So what happens if we infect the mouse with prion protein, but also give the mouse the ASO? And this third pair of pictures shows a mouse 169 days after prion infection, so about the same time point as the row above. But this mouse received the misfolded prion protein as an inoculation and also received a single dose of ASO at day 120. So we waited until the mouse showed symptoms of disease, gave one dose of the ASO, and then looked at the brain tissue 49 days later. On the left, you can see that there is some clumping of prion protein, but not nearly as much as in the infected but untreated mouse, the picture right above it. And on the right, there are some vacuoles, but far fewer than in the untreated mouse, one picture above. So there's definitely some benefit, at least when we look at the tissue. We also look to see what would happen if we gave the ASO earlier. So that's this last pair of pictures. We have a mouse at 161 days after the prion infection, so also about the same time as the mice in the rows above. But this mouse received a dose of ASO before being infected with misfolded protein, and also doses of ASO at day 46 and 106, days after infection. So here the results are even better. There are very few clumps of prion protein, and there are very few vacuoles. So these experiments suggest two things. Use of an ASO targeting prion RNA seems to prevent or reverse some of the pathological findings that are associated with prion disease. And there seems to be a benefit if you use the ASO prophylactically, that is before infection, as in the bottom row, and also a benefit, though perhaps less of a benefit, if you delay treatment with the ASO until after the mice are visibly sick. That's that second row up from the bottom. So the next question is, does the mouse actually seem better? It's nice to reverse evidence of disease pathology in brain slices, but is the mice actually better? And the short answer is yes. So here's one slide with a lot of data. We have body weight, top left, nest building, top right, and survival on the bottom. As I said before, both mice and humans lose weight during prion disease, so we can follow body weight as a marker of disease progression. The nest building in the top right is a useful thing to measure because healthy mice instinctively build good nests. When you see a mouse not building a good nest, it tells you that the animal's sick. Maybe it's cognitively impaired, maybe it's physically impaired, but there's definitely something going on. And on this scale, 2 at the top is a normal nest and 0 is the worst score. And of course, we're always looking to see how long the animals survive. That's the mortality plot on the bottom. In this particular experiment, if you look at the top of the mortality plot, you see some triangles. We delivered a dose of ASO prior to infection with misfolded prion protein and a second dose around day 80. The gray lines on all plots are mice that didn't receive any ASO. So if you look top left, you see the doses, and the gray line represents 0. It was just a vehicle delivery. And then as we increase from pink into darker colors to the dark purple, the amount of drug, the amount of ASO delivered increases. So again, if we look just at the gray lines, this is what happens to an infected mice that doesn't receive ASO. The top two plots start at day 120. So remember, that's 120 days after the prion infection, and it's the point at which the animals are showing outward symptoms of disease. And in the body weight plot, you see that the animals who didn't receive the ASO, the gray line, quickly lose weight. And then the line ends around day 150 because the animals have actually all died by day 150. As we increase the amount of ASO delivered from light pink to the dark purple, the animals are looking better and better, both on body weight and on the nest building scales. And on the bottom, you can see that the animals that received ASO are living longer. So this is one of many, many experiments. Like I said, I can't possibly do the preclinical work justice, but they all have the same general findings that using an ASO to reduce the amount of prion RNA, which then reduces the amount of prion protein, is beneficial to the mice. And there's benefit in initiating ASO treatment even after the mice show symptoms, but there's a greater benefit if we introduce the ASO earlier. Experiments like this one and many, many more gave us the confidence that we needed to proceed with the program. So next steps. We'll continue our research collaborations and we'll continue to generate data to understand the effect on animals. The more we learn in animals, the better we're able to plan for human clinical trials. We like to shift our risk to the animal studies to protect our patients. We'll complete the ongoing toxicology studies, which, like I said, are a sort of standard set of animal studies that regulators expect to see before giving permission to enter clinical testing. We're moving full steam ahead with clinical trial planning. We've been working with several experts in prion disease to learn all we can about the disease and the challenges we might encounter in clinical trials to understand the population as well as we can. And we'll meet with FDA and regulators in other countries to discuss the types of clinical trials that should be conducted to generate the evidence that we need to support eventual approval of an antisense drug. So finally, quickly, I'd like to send out some acknowledgements. First, a big thank you to the CJD Foundation for inviting me, and a big thank you to the patients and families who participate in research in the past, who are considering participating in research in the future. I can't overstate how important it is to drug development that there's a thorough, documented understanding of the natural history of the disease. If you've ever volunteered for a clinical trial or consented to have your or your loved one's medical history used for research, I can almost guarantee you that you've helped to advance the development of new drugs. Let me also thank the people named here. We have lovely, productive collaborations with individuals at the Broad Institute, the McLaughlin Research Institute, and Rocky Mountain Labs. We're working with a stellar panel of clinical advisors to shape the clinical trial plans. And here are the colleagues at Ionis who contributed in some way to the work and activities that I showed you today. It's a great group of dedicated scientists who are motivated to bring treatments forward to patients in need. The overall Prion team at Ionis is actually much bigger. It includes patient advocacy and manufacturing, quality assurance, project management, statisticians, operations, and many others who are working to ensure success as we enter into the clinical phase. I understand we'll have a Q&A session next, but if your question doesn't get answered or you have additional questions or you're just curious what's going on, please reach out to us at the address we show here at the bottom. And I'll stop there. I hope you have a better understanding of the work we do. I hope I've conveyed how excited we are to be working in prion disease and how grateful we are to have you engaged and following the work. So thanks again to the CJD Foundation for the invitation, and thank you all for your time.
This is very exciting. This is the first time that there will potentially be a clinical trial that's sponsored by industry, which is a completely different ballgame compared to having government or independent investigators do a clinical trial. It also is a little bit different compared to other prior clinical trials in that it's looking at a different mechanism. So most prior clinical trials looked at blocking the conversion of the normal prion protein to the abnormal form of the prion protein that causes disease. But as Anne was mentioning in her talk, this approach would remove the fuel to the fire of prion disease, essentially removing everything that it needs to continue to happen or to even start potentially. So the first question we had, Anne, if you'll bear with me asking it again, is, will ASOs work for all types of prion diseases? Will they work for all types of mutations?
Right. So the short answer is yes. A single drug should address all types of prion disease. And that's because, as Brian says, we're aiming to reduce the fuel for the fire. We'd like to knock down the amount of normal prion protein. We just run-of-the-mill prion protein that's circulating in everyone. We think if we can lower that, at least the animals tell us, if we can lower that, there's less fuel for the fire and it helps protect against disease, or it helps with disease. So yeah, we're in a good situation here where we were able to design the ASO to bind to a spot on the RNA that's distant from any mutation, and it should address all forms of prion.
Wonderful. And there are a lot of family members in the audience, and I know they're all asking, when do you think clinical studies are going to start in humans?
Yes, I expected that question. So as I described, we're currently conducting a series of studies in animals that need to be completed before human trials can start. We're also in the process of engaging regulators to discuss clinical plans, assuming that the data collected in the animal studies is sufficient to support the initiation of clinical testing, and assuming those conversations with regulators proceed well. Those are ordinary steps of this process. We're used to doing those two things. If that all goes well, we anticipate a clinical start in the second half of 2021, so second half of next year. As details about the clinical plans take shape, we can keep you informed through the CJD Foundation and other avenues, posting on clinicaltrials.gov, for example. I can't share details of what those trials look like today because we haven't decided what those trials look like today, and I wouldn't want to tell you something today that might change tomorrow. But I can tell you we are actively in the process of designing those trials, talking with experts, talking with regulators, so that we're ready to go as soon as the drug is ready to go.
It's excellent news. Are there going to be multiple treatment sites? Is it just going to be in the US, other countries? Is that something that you even know yet?
Yeah, I would expect this will be global. We have relationships around the world, and we have an interest in being efficient. When we design these things, and really the design of experiment is critical here. We want to do the right trials. We want to have them sized appropriately. We want to have the duration of the trial appropriate. So we answer the question as quickly as possible. I like to get things done. So we will intend to use sites around the world. We'll scatter them geographically to help individuals get access to them. And that'll also help us answer those questions as quickly as possible.
And ASOs have been used in other neurologic diseases like spinal muscular atrophy. What do we know about the side effect profiles of ASOs in general?
So ASOs, yes, we probably have 25 drugs in the clinic right now, some of them intrathecally delivered and some not. So we have experience with sort of class findings, and the IT-administered ASOs really are pretty well tolerated. You can have some side effects from the intrathecal procedure itself. That's actually more common than a side effect from the ASO. But it's important to know that each ASO is different. They're built from the same building blocks, so there are a lot of similarities, and we can anticipate similarities across programs. But each ASO has a unique target. Here we're targeting the prion RNA. So we need to get through those critical animal studies now to see what the side effect profile looks like in animals. These are called toxicology studies. And again, this is a standard set of experiments, but what you do in those is you dose the animals much higher than you intend to dose people, and you look to see what could potentially happen. And from findings that we'll get from those studies, we'll learn better how to monitor patients in clinical trials.
And the target of your ASO for prion disease is the normal prion protein, which is something we all have. And assumingly it's something that we all need, or is somewhat important perhaps. What do we know about any potential effect of blocking our normal prion proteins?
Yeah, so here's another place where we're really pretty lucky. We know from knockout animal studies, so where you completely eliminate the amount of prion protein from conception, that the animals are viable and they grow and they're healthy. The only side effect that you see in animals when you completely knock out prion is a peripheral neuropathy, a mild tingling in your fingers and toes that we're unlikely to see in the clinic because we're not delivering the drug systemically, we're delivering it into the intrathecal space. So it's going to be circulating in the CNS, And it will be cleared out through the periphery, but by the time it gets out there, the doses really are so low that we wouldn't have much knockdown in the periphery. If we needed to knock down prion in the periphery, this is not how we would do it. This would be a really inefficient way. So we will monitor. That's a good example of something that we'll monitor in the clinic to see if there are any effects like that, but we wouldn't expect them. We think we have pretty free rein to knock down prion in the CNS. Another thing I didn't mention in the talk is ASOs are not a good choice if you need to eliminate a protein completely. Invariably, some of the RNA is going to get to that ribosome and a protein is going to be made. We can't knock it out completely. So there will be residual prion regardless of the dose we choose and the regimen that we use. So we're not even mimicking those knockout animals. But as I said, even if we get it down very low, we wouldn't expect any effects due to the knockdown.
And I believe Eric Minical also perhaps even presented data at one of the Foundation family conferences, is that we do know that there are at least some individuals that only have one copy of the normal prion protein gene. So they're not making as much normal prion proteins as the rest of us, and they seem to be okay. And that's encouraging as well.
Yep, good points.
I just wanted to remind people that this is being recorded and the CJD Foundation will post it on their website. So if you didn't catch anything or you want to go back and look at it, it will be there. Also, we are collecting your questions. We're prioritizing ones from the family members, of course, but any questions that we don't get to, we'll make sure that Anne will get, and we'll get you an answer. So one thing that I know is that there are a few family members here who have loved ones who are actively ill, and some may be in later stages of the illness. In this type of study, would you include people at different stages? And if so, what type of effect might you expect from someone in a late stage of an illness?
Yeah, that's a difficult question, and that's a detail of the design of experiment that I can't answer today because we haven't decided there yet. It is true that we're pursuing what I consider two populations here. So we are encouraged by the data in animals, both as a prophylactic use in not yet sick inherited prion disease and as a symptomatic treatment in people who have the disease. What that trial will look like, inclusion, exclusion criteria, I can't really speak to today. I don't want to be wrong tomorrow. Obviously, the earlier the better in terms of treatment having a chance to work, but that doesn't mean that the trial itself would be necessarily very restrictive in terms of eligibility.
One thing that we saw recently, which Dr. Simon Mead talked about not too long ago in regards to a monoclonal antibody against the prion protein, is that they couldn't get a proper clinical trial funded, so they used the treatment compassionately. Can you talk to us about any compassionate use case scenario for this drug? Is it all going to be through clinical trial?
Yeah, that's another really difficult question. I can say we are all in as a company in doing the proper clinical trials. We're excited about this opportunity, and we're thrilled to be innovating in this space where there is such a dire need. So the clinical trials are the first priority. And again, designing trials and executing them to generate evidence about the drug's safety and its efficacy, and doing that efficiently and as soon as we can. That said, clinical trials by their nature are in a limited number of individuals. So we're also thinking about the people who won't be enrolled in trials for any reason. Maybe you don't live close enough to a site that's running the trial. I don't have the answer right now. I can say we are working through how best to address the needs of those patients also. But we need to get into the clinic, and we need to get a handle on the pharmacokinetics of the drug. You know, we need to figure out our dosing regimens. We need to see the safety profile in a way that we can interpret it and understand it before we could consider something along the lines of a compassionate use.
As you said in your study, there are multiple different phases for drug trials. There are phase 1, phase 2, phase 3. Some focus on whether or not the drug is harmful, Another focus on the dose that you need to be effective. And then, of course, part of drug studies is whether or not it's effective. So you still have a fair amount of those things to go through. And not knowing exactly how you're going to be conducting the studies, what would you say is a reasonable time frame for knowing whether or not this would be an efficacious treatment in humans?
Yeah, so you're right, there are these phases, but we're not bound to them, as I mentioned, and we'll try to answer as many questions as possible in a trial to speed things up. That said, drug development is not a fast process, so you should expect it'll be several years before we can say definitively if this drug is safe and effective.
And then I think you mentioned in your talk that these ASOs will have to be given repeatedly. Is there any concern about being exposed to ASOs multiple times?
Occasionally you will see antibodies to the ASO develop. This is not an uncommon thing. We've not seen neutralizing antibodies, which means they never seem to matter. Your body's recognizing something unusual and it's kicking out antibodies to it. But as long as they're not interfering with the ability of the ASO to do its job, and they're not themselves causing side effects, then we don't really worry so much about it. So it hasn't been an issue in other programs. This is one of those areas where the similarity from one ASO to the next gives us some comfort that we wouldn't see that kind of a problem here either.
Many people in the field envision that the treatment of prion disease would resemble what we see with HIV. So in HIV, the virus can mutate and adapt to the treatment. And there's some evidence, a lot of evidence, that prions can do the same thing, that they can become resistant to different therapeutics. So the thought was that if you had to treat prion disease, you would have to use a cocktail of different anti-prion drugs. Now, again, this is a different approach that you're taking because you're not looking at blocking the conversion of the normal prion protein to the abnormal prion protein. But do you envision possibly using dual or multiple therapies with ASOs as a possible treatment?
So the therapies where you worry about that binding tend to be the ones that act on the protein itself. So we talked about gene therapy acts on, so I should go this direction, gene therapy acts on the DNA and ASOs act on the RNA, and then small molecules or protein-based drugs act on the protein itself, where it is really critical that, generally, it's really critical that your drug binds well to the protein. So the conformation of the protein, the way it's folded or misfolded, can affect how effective your drug is. And that's one of the challenges in protein misfolding diseases is we often don't know what the regular formation folding of a protein is, let alone how many myriad of different conformations it might take when it's misfolded. So it can be a challenge to find something to bind to the misfolded protein. We're working on the RNA. The RNA is always going to be the RNA, and we can constantly keep hitting it and knocking it down, we think, which should, as you say, lower the fuel for the fire. I hope that drugs will also be developed acting on the proteins that are effective, and it might be down the road that we learn that a cocktail of those is even better than one or the other. But in the meantime, I think we can pretty safely say we're going to hit the RNA. There won't be a mutation issue. There won't be an evolution issue like you worry about in HIV. We should be able to consistently knock that down.
And it sounds like people are going to need multiple treatments of the ASOs to knock down the RNA for the protein. How long will people have to be on this?
We don't know that yet. So you do have to think that we're not changing the DNA, which means the body is going to continue to make copies. It's going to continue to make RNA of the prion protein, and we can give an ASO which will destroy it, but as the ASO wears off, then those RNAs won't be destroyed and the protein will be made. So one of the important things that we need to work out, and the animal data that we're generating now is going to help us with this. We need to work out what the drug regimen is. How frequently do we need to give it? What's our target knockdown? How much do we need to give with each of those doses? And come to a regimen that is tolerated from the patient perspective, but also effective in terms of having enough drug on board to keep the fuel down. I really like that analogy. So it's something we'll work out in our animal data, and we will put it into clinical trials and test it and see if the drug is acting the way we expect it to act. In terms of longevity, like how long would you have to stay on this drug? That's another thing we don't necessarily know. And that's a difficult question to answer. It's hard to conceive of a trial to answer that question. For prophylactic use, I feel like we're going to be developing a regimen that is going to be used long term. As long as disease onset hasn't hit, we think the drug is responsible for that. I would think you're going to keep taking that drug for as long as the patient is interested in continuing to take the drug. For the symptomatic population, it's going to be really interesting to see if we can arrest the disease and work our way into some sort of maintenance regimen that's tolerable for long-term treatment. I think that's likely where we're going to end up, but we'll have to see what the data tell us.
And as you know, because you've met the foundation and many of the family members, we have a pretty dedicated group of patients and families that really will do a lot to contribute to research. And of course, the way that this drug is administered is, what we call intrathecally, so through the spinal column. I think most of our families, just for the opportunity to participate for a potential treatment, that's not going to be an issue, but for some it may be. So can you talk about, is there any way to get around that in the future?
Maybe. So there's work being done on that. We're working, and others are working on ways to change the ASO so that it can cross the blood-brain barrier. There are some cool ideas out there. I don't know what will pan out, but yeah, intrathecal delivery is working well, and it is generally well tolerated. And I think you'll find that it's scary at first, but it really isn't that bad. I've heard that it's the tolerability of that first intrathecal injection has a lot to do with the person who first talked to you about it. If you're introduced to the idea by somebody who thinks it's scary, you're going to go in thinking it's scary. If you're introduced by a medical professional who knows that it's an injection, and we do this all the time, the anxiety level can be a lot lower. But that said, yeah, as we move forward, we're going to have more and more drugs delivered by this mechanism. We will look for ways to make the tolerability better and the appeal better for the patient population.
One of the things that I'm sure you and Ionis has talked about is, how do you set the stage for such a study? You have two populations that you want to treat, those who are at risk. So you have to identify those who are at risk. Then you have those who are actively ill, who unfortunately have a very rapidly progressive illness. So what are some of the strategies that you've contemplated in addressing those issues?
In terms of sort of preparing the community, that kind of thing?
Yeah, so to make sure that you get the study population that you need for these studies.
Yeah, so there are approaches we can take with education in the medical community, encouraging early diagnosis, letting physicians know that these trials are coming, and if they could be on the lookout and try to think prion disease earlier in the diagnostic, that would be helpful. And connecting physicians with Brian and others who run these surveillance centers, so we can work from that side to get the word out and encourage early diagnosis. And then we also we like to work with the patient community through the CJD Foundation and others to talk about what it's like to participate in research, to make sure that somebody who might be interested in participating in research is connected with physicians who are involved and knows the opportunities that are available. And as we build up to this, I should mention also, it's not just the next time you hear from us will be trials. There are opportunities that we'll have to help with research in ways other than clinical trials, like filling out surveys. We want to understand the needs of the patient population. We want to understand what you're expecting from a drug so that we can try to deliver it. So we're right now talking with the foundation about collaborating on a few community-focused initiatives that can help to support drug development, our drug and other drugs for prion disease. I don't think there are plans set yet. I would imagine we might have a webcast later in the year to share those opportunities, but that's also near term for us, is helping the community be ready to know what's coming and participate along the way, so that, again, the design of experiment, the trials that we do are meaningful and useful and answer the questions that need to be answered. The more we know from the community about the disease and your lives and how it's affecting you, the better we can design those trials.
And I have to say, obviously, Ionis has a stake in letting people know that they have this upcoming study, but Ionis really has already contributed an awful lot to the field. They're a sponsor of the CJD Foundation Family Conference, Strides for CJD, and even the International Prion Conference. And I think that really speaks volumes that they really care about the field itself, and they want to make the situation better for everyone. And of course, they would love to do it with the pharmaceutical, but they're also looking at other ways to help the patient community. And I think that's appreciated. Some of these people that would potentially be in your study and are people that are young and maybe of childbearing age. So is there any contraindication with pregnancy for ASOs?
So that is part of the standard set of toxicology studies that are done to understand if the ASO crosses into a fetus or into breast milk, and that's a set of studies that's ongoing now, so I can't answer that question. I can say that with other ASOs, there have not been contraindications. Often in the course of a clinical trial, we ask you to please not get pregnant because that's complicating. But once the drug is on the market, once you're just doing long-term chronic use, the data will tell us. I can't say that there won't be a contraindication. I can just say that it generally doesn't pop up with ASOs.
There's a couple other questions, and I think we'll end for the night. One questioner said that they think that there are some ASOs that are used to treat one or more illnesses. And if that's the case, have there been any major lessons learned from that experience?
ASOs to treat one or more. So I'm sorry, Brian, do you think approved drugs, like lessons from approved drugs, or really an ASO that's going to treat more than one disease?
It sounds like the question is really asking about an ASO that would treat one or more illnesses.
Yeah, so it's possible. There are certain proteins that are implicated in more than one disease. I can think of a particular protein that's implicated both in ALS and frontotemporal dementia, or a dementia state, where it might be valuable to knock down that particular protein for both of those patient populations. So that works well. There's no reason we can't address both of those patient populations with that one strategy, not unlike what we're trying to do here with addressing really two different patient populations, two different needs, with the strategy of knocking down the same protein. We do value the specificity of ASOs, that we are targeting a single protein, and we can do that in a way that doesn't inadvertently knock down a bunch of other proteins that you might really need. So we value that. But yeah, there are situations where the same protein is problematic in more than one disease.
There's been some data to suggest that the normal prion protein might be involved in the neurotoxicity seen with Alzheimer's disease. Has that been discussed at all?
Probably not a good tangent for us today. There's mixed opinions out there about that. first and foremost, we want to knock down prion for prion disease.
The reason why I ask is because oftentimes when we try to advocate for funding, we as a prion community often have to tie ourselves into those larger communities. So that was the basis for that question. I didn't mean to put you on the spot.
I understand. I understand how grants work. I said it earlier, we're all in on prion disease. We're not going to go off on those tangents. We want to address this first.
So what can the families that are listening do right now to help you in this endeavor?
Yeah, I think the important thing now is, as I said earlier, just get connected with physicians who are involved in research. If you think you want to participate in clinical trials, make sure that physicians know that. And the CJD Foundation can help you with this. They can connect you with physicians who are active in research. And beyond that, just look for announcements from the CJD Foundation. Like I said, we're going to start some initiatives of just asking you questions about what you would want from a drug, and we'd love to get as many responses as possible so we know we're heading in the right direction. So really connect. Everybody here knows the CJD Foundation. That's why you're here. So stay connected here, and Debbie and the good folks there will help you know when there are opportunities to help.
And some of our families also are enrolled in the Prion Registry. Would that also be an avenue that they could discover the trial and details when it comes out?
Yeah, I'll admit that I don't know a lot of details about how the Prion Registry works. I think there's an effort to make it a two-way communication. And if that's successful, meaning that the registry can blast out information to you, then yeah, we'll absolutely make sure that's another avenue where we're sending information. But it'll all be the same information. If the CJD Foundation knows something and gets it out to you, the registry will know that same thing and also get it out to you. So you don't need to check multiple.
Any ending words for family members or colleagues on the call?
Oh gosh, I should have prepared for that. I can't not thank you again. The involvement, the awareness of the community, the support has been really important, and we're working on this for you. So I want to thank you for being engaged with us and helping us along the way. It should be an exciting next several months and several years, and I look forward to staying connected.
Prion, I wanted to thank you for joining us tonight, and thank you very much for your work in developing a treatment in prion disease and really being a good steward for our field as well. You've already contributed a great amount, and I know I speak for the families saying that we're greatly appreciative. So thank you very much.
Thanks, Brian.